today what I want to talk about is investigating the effect of the plasminogen system on the brain endothelial cells. So to start off with, we have another version of everybody's favourite diagram, which I won't go into a lot of detail. But effectively, you've got plasminogen that is inactivated by TPA into plasmin, and that should cleave the blood clot. And obviously, we're all familiar with this. This is something that's been gone over a million times. However, particularly in the context of stroke, one of the main things that actually ends up causing pathology is breakdown in this endothelial layer. And that then allows a whole lot of things and blood proteins to enter the brain and do damage to the neurons below. So my question really was around plasmin itself. Does plasmin affect those endothelial cells? And there's been quite a bit of work in this area previously. It's been shown that plasmin can activate MCP1 and the matrix metalloproteases, and in turn, these can trigger barrier loss within the brain. However, surprisingly little is known actually about the direct effect of plasmin on the endothelial cells themselves. So that's the question that I really went out to look into. So just a quick overview. First, I'm going to talk about how I monitor these plasma-mediated changes in real time. Then secondly, show you a bit of ICC and tell you about the junctional molecules and how they're changing. Thirdly, I'm going to show you some drugs that we've recently screened. And then finally, just a few future directions and new toys that are coming on stream. So how exactly does one monitor the plasma-mediated changes in barrier function? Well, as I'm sure we're all aware, stroke and the interactions of the plasminogen system are really highly dynamic, and so it would be really good if we could track these in real time. So that's where this technology, electrical cell substrate impedance sensing, or ESIS, comes in. What ESIS is, it's a way of measuring the endothelium barrier integrity. So I generally work with these HCME-CD3 cells. They're an immortalized human brain microvascular line that's really well used, really well characterized in the field. And then we grow these on these 96-well plates that look like nothing special to the eye, but they actually have fabricated onto them these little gold electrodes that make these indigitated fingers. And this is really useful because it means we can put tiny electrical currents through. So if we cut that well vertically, you can see we've now got the endothelial cell sitting on top of the gold electrode. And then we can put a tiny electrical current through here. And this current is so small that the cells cannot perceive it. But it's large enough that we can attach an ESIS machine, which is effectively a $100,000 glorified voltmeter, and it can then measure the resistance of each of these wells, and it goes around and clicks its way around every 15 minutes. And because this is very non-invasive, we can do this continuously over the course of days, and we can watch really carefully what's going on with plasmin. Now, as I'm sure we're all aware, plasmin is difficult to study in any system that contains serum. Because serum is obviously a blood product, and so it's going to contain the plasminogen system, all its regulators and everything that interacts with it. The problem with brain microvascular cells is they're actually quite a delicate cell type. Even though they form these really tight junctions, unless they get all the support they need, you lose that barrier very quickly. They're generally grown in fetal bovine serum because, again, that's close to the plasma that they would have within the body. And that's really important for maintaining barrier and function, but we can't use it in the system because it's just going to inhibit any plasmin we put in unless we put in absolutely boatloads. So what I've developed is a novel serum-free conditions which were developed, and basically I went through and screened a whole load of different formulations and came up with something that was both able to maintain the barrier integrity and didn't interact with or contain the plasminogen system or any of its activators. I did that using a whole load of fluorescent substrate cleavage assays. Now, whilst this gives us a less physiological model, we've removed a whole lot of the safeguards in there, it gives me really precise control. I can put in exactly the amount of plasmin, plasminogen, TPA, whatever I want, and know exactly what is on those cells. So that allows me to answer this question of how does plasmin actually affect the endothelial cells? And the answer is it's not good. So just to orientate you with this graph, on the y-axis here is overall resistance. You want that number to be high. The higher that number, the tighter those cells are sticking to each other, and the less would be able to go through the brain. The first thing you'll notice is the spike in every condition, including the control, which is the blue here. This is just a feature of ESIS. When you take a cell out of the incubator and you muck with it, it doesn't like it, and it transiently increases it. Now, normally you wouldn't see this on an EVOM or a chopstick type electrode. They're just not sensitive enough to do that, and they don't have the time resolution. But here you can see that's happening with ESIS. What is really intriguing, though, is that immediately after it comes down from this point, we get a concentration-dependent decrease here. Again, these are twofold steps, up to 640 nanomolar plasma. And you can see that we get this concentration decline that's almost immediate. However, intriguingly, and this is not something you would see unless you had this real-time data, we then get this kind of plateau that lasts roughly 4.5 hours, which is kind of the magic time point for TPA treatment, which to me seems a little coincidental to not be something interesting going on. Coming on from that 4-hour time point, you can see that we then get this secondary decrease, and it falls, particularly the 640 and the 320, below the control there. Now, plasmin's another one of these things which is really difficult to work out actually what the concentration on the surface of an endothelial cell with an intact brain is, but even at half or quarter of that concentration, we are getting statistically significant differences. Which brings you on to my next question of, is this due to plasmin's enzymatic effect, or is this something else? And so to work that out, I added the inhibitor of plasmin, alpha-2 antiplasmin. And the results were not unsurprising, but quite intriguing in that, so here's your control in the blue and the plasmin again. The alpha-2 antiplasmin itself was actually protective, which, given some of the data we've seen this morning, is potentially surprising. But also this plasmin alpha-2 antiplasmin complex is sitting directly on top of the plasmin. So if you kill the plasmin activity, you're not getting this effect. So there seems to be something enzymatic going on with plasmin. It's not anything else in the preparation. And the final purified protein I'd like to show is just TPA. Again, much debate as to what effects of TPA are TPA and what is its downstream. But this is a serum-free model, so we are truly looking at TPA here. And this is ultraplays. It's just we got one human dose that's lasted us the last 5 or 10 years. But what we've got here is the TPA, and you can see that there's absolutely no difference there at all without plasminogen. And whilst I don't have the time to go into it today, we've done surface-bound plasminogen, we've done a little bit of work in serum, and again, we start to recapitulate those effects. So once you add that plasminogen back in, you start to get the damage again. So the conclusion from this really is that plasmin causes a reduction in brain microvascular integrity over that time course. So the next question I wanted to ask is, ESIS gives us this lovely functional measure, but I'd like to know what's going on a molecular level. To do that, I did ICC. And again, because we've got this ESIS data, we can very precisely tell you where to look. And so we decided to look at 4 hours, that being clinically important, and at the end of that plateau, and then at 12 hours. So just to show the first of these stains, you can see here this is VE cadherin, and that's one of the key cell-cell adhesion molecules responsible for maintaining barrier integrity. So 4 hours after plasma addition, you can really see there's a substantial decrease there, and after 12 hours, similar. And even what remains is actually distributed quite differently. It's not as focal, and it's difficult in these to see where the outlines of the cells are. And of course, ICC has immense variation, and as we saw yesterday evening, you can just pick the bit you like. So what I've done is I've gone and done high-throughput imaging using our operator system. So each of these is 8 fields in each of 3 independent experiments. And then I've quantified it with vJuncture, which is some software in R that I've written that does this entirely without intervention, so it's much more holistic. And you can clearly see here the VE cadherin expression is decreased a little bit at 4 hours and more at 12 hours. And whilst there's definitely some variation across the monolayer, this comes out really quite significant. The other nice thing about quantifying it is I can condense hundreds of images down into a single heat map. And what was really interesting here was that 4 hours after plasmin addition, we saw this massive decrease in beta-catenin. Now, that's interesting for 2 reasons. Firstly, beta-catenin is a key molecule that sits between the VE cadherin and the cytoskeleton of the cell. So it actually is maintaining that junction. It's also on the Wnt signaling pathway. So the fact that it's being lost from the junction says it's going somewhere, it's doing something. Interestingly, partial recovery 12 hours after plasmin addition. So I then looked at a whole load of other, the V-cadherin we discussed before, but Claudin 5, PECAM1 are also seeing decreases that again increase towards 12 hours, and zonocludin 1 again increasing. And this is from a large number of drugs, sorry, not a lot of drugs, a large number of images, and I've just had an honest student who's then repeated all this and got very similar results, which I've found strangely comforting. So my next question was, can we use any drugs to block this effect? And so when I was trying to write up my PhD thesis, I thought, let's do a quick summary diagram of plasmin signaling. It can't be that complicated. It is that complicated. So as you can see from this, plasmin alone, it's a highly promiscuous protease. It hits everything from TPA, MMPs, MCP, and these then hit their own receptors, and those receptors then hit the junctional molecules. And as I showed on the previous slide, it's not one clear junctional molecule. So even if this was one singular pathway for each junctional molecule, that wouldn't be of any help. The nice thing about working in a serum-free model is this is simplified, but still there's good evidence that all these factors are present. So the obvious linchpin here would be get rid of the plasmin, but as people have said earlier today, that's not something we can do, because if you don't have the plasmin, you don't clear the clot, and the part of the brain dies. So that's not an option. So we need to go in somewhere else. But the signaling is highly integrated and highly complex, so there's not a smoking gun. However, fortunately for me, there's a whole load of drugs that exist out there, and you can buy off the internet and screen. So that's what I did. Unfortunately, the IP people have said I'm not allowed to tell you what the drugs are, because some of them have worked. But I am allowed to show you the underlying data. So the way I presented this data, 0 here is the effect due to plasmin alone. So anything below 0, the drug has actually made this effect worse. Interestingly, though, 100% at the top here would be a total rescue. So that is, plasmin is now not in any way affecting the barrier. What is really interesting is drug number 11 here. We're getting 74% protection, which is quite substantial. But the other thing that is really interesting, and the takeaway of this, even though I can't tell you what they are, is that these drugs target multiple signaling pathways. And even of these positive hits, they're not all hitting the same thing or different places on that same pathway. So, as I was saying in the previous step, this is a highly integrated system. Also, interestingly, some of these things that people have suggested in the literature might be protective, we're actually seeing the opposite effect in these cells. So from a scientific point of view, this tells us that various pathways are probably contributing. Now, what I've shown you here is a single concentration of each of the drugs at a single time point at 12 hours, but each of these drugs we actually ran at 2 concentrations, and we have the real-time data. And whilst I don't want to drown you, this is drug number 11, which was our best hit. And you can quite clearly see here that you've got your vehicle in the red and your plasmin in the blue. Interestingly, this drug has very little effect on in the early period where TPA is effective. However, if we drag that out to 10 hours, when we're starting to get well past those deadlines where TPA will be contraindicated, at the lower concentration, we're seeing protection. At the higher concentration, we're almost running exactly on top. So I then asked the question, have we destroyed the plasmin? And we haven't. I've checked. This drug has no detectable effect on plasmin's activity. So this is actually blocking a signaling pathway downstream. And then I had a great honours student this year who's gone through and repeated all the ICC. And again, I'm showing you here percentage protection, both at 4 hours and 12 hours. And you can see across the board we are seeing protection, particularly in the PECAM. Less protection at 12 hours. Kind of difficult to say what's causing that. But the big takeaway here is, regardless of where you look, this drug is a hell of a lot better than nothing. So where to next? Well, the great blessing and the great curse of this model is that it's a very purified model. It is not trying to recapitulate what's going in the body. But obviously, that's where we'd like to head, back towards more physiological stimuli. The first of these is shear stress. So obviously, an endothelial cell in the body is constantly being perfused by fluid over the surface. And that causes the cells to tighten and really causes these cells to maintain that integrity. And it also causes the removal of non-surface bound plasminogen system proteins. So I've done some provisional things with alpha-2 antiplasmin and plasminogen and all the complications. It gets very in-depth, but happy to chat further if anyone would be interested. But I think really a shear stress, a reperfusion model would be great. This is obviously technically challenging to do, so I've done the Kiwi thing and built my own thing. So what we've got here is an off-the-shelf syringe pump, and that goes forwards and backwards and produces reciprocating flow. Obviously, that's not the least bit physiological, so you put some valves in a circuit, which is exactly the way your phone charges, AC to DC conversion, and that, the video's gone a bit jumpy, but basically what that does is that converts this reciprocating motion into unidirectional flow. So you can see the liquid in the reservoir. This is sped up about 20 times. You can actually see it going up and down, and regardless of the colour coming through, it's always moving the same direction over the cells. Big advantage of this, it's cost effective. I can do this for about 20 American dollars, 30 Kiwi each. You can reuse them. If you buy a similar system from a biddy, you're talking 300 New Zealand a system. The other nice thing about this versus a lot of the microfluidic stuff is this is big enough to tinker with. You can sit there and fiddle with it. The latest thing I've done is actually put little Lua locks in it, so we can just inject drugs while it's going, exactly as if it was a patient or a mouse or something. It's also ESIS compatible. And finally, because this is so cost effective, you can hook 6 of them up and you can do parallel studies. In fact, I've just bought the bigger rack, so now we can do 12, but it becomes a lot of tubes. So this is what it looks like. This is 2, and it's a lot of tubes, so you can imagine 6 is a lot of tubes. But basically, this is the pump, these are the tubing runs, and the ESIS chip that then the machine sits on top here. And this whole thing is spec to sit inside an incubator, so that gives you your CO2 and your temperature control. It's all quite a nice, neat little system. Finally, this is the ESIS data pushing out to 20 hours. This came out of a grant we submitted recently, but I've now got this out to 4 or 5 days. And you can see that the shear stress is causing this increase that after a day or so does plateau out. Interestingly, we're also seeing our remodelling, which, again, other people have shown. You're going from this kind of not very organised to a much more stress fibre-oriented sort of a phenotype. And again, we're seeing remodelling in the VE kit here. Other things I'd like to add in. I'd love to do oxygen glucose deprivation. Currently, this is not really a stroke model. It's more of a plasmin model, and obviously we know that OGD is going to have all kinds of effects on the metabolism, as we've heard this morning, but also the MMPs and all kinds of other things. So I've just taken custody of the new hypoxic incubator and done the math to check I'm not going to hypoxic the students. But then we can mix this with a serum-free media that's formulated glucose-free and get us much closer to a stroke model. The final thing I'd love to do is work back into serum and plasma samples. So still working on the technical details, so if anyone has any ideas, that would be great. Thinking maybe start with healthy donors, TPA treat them. We can do stoppage of flow. We could maybe make a clot. There's a million combinations, so I'm just kind of trying to pick where to do the best things here. The other lovely thing about this is because this is a live system, if there is anything going on with inflammation, these cells will respond to TNF-alpha, IL-1 beta, any of the inflammatory factors, any of that, you'd see it, whereas you might not if you were looking at single proteins. So very quickly, some conclusions. Plasmid alone causes substantial and time-dependent, just jump spelling-wise, anyway, reduction in the brain microvascular integrity. A variety of drugs can ameliorate this effect. And finally, we're going to add some more physiological stimuli. So I'd just like to thank Catherine Angel and Scott Graham, who have been my PhD supervisors and now mentors on this project, as well as the rest of their lab and our funders in New Zealand, as well as the university. Thank you very much.